Hi, good morning. Last class we discussed the basics of bryophytes. In this video class, I like to explain the topic Rothmeiler and Proskar system of classification. We know that bryophytes are called the amphibians of the plant kingdom. That bryophytes are classified by different scientists based upon their views. So here we are going to discuss Rothmeiler and Proskar system of classification. According to this classification, bryophytes are classified into three classes. That is, first one is called hepatic opsida, that is liverworts. And second one is called anthocerotopsida, that is called hornworts. And third one is called mosses or bryopsida. So that is the three order of bryophytes. And when we move into the not order, these the three class. When we move into the order of this bryophyte, the class hepatic opsida possess or divided into 7 orders. That is Tachachiales. In this slide, you can see the left one. You can see, look at my cursor, you can see the left one. Tachachiales, Calobriales, Jungermanniales, Metzeriales, Marcantiales, Spirocarpales, and Monocleales. These are the seven order involved or coming, which comes under the category of the class Hepaticopsida. But when we move into the second class, that is Anthocerotopsida, you can see that Anthocerotopsida can also call hornworts that possess only single order, that is called anthocerotales. If you look at here in the center of the slide, you can see anthocerotales. When we move into the third class, that is called Bryopsida, or we can say it is called mosses. It is divided into 5 subclasses. 5 subclasses. In first category, you can't able to see the subclass, but here you can see the 5 subclass. That is called, first subclass is called Spagnidae, Andreidae, and third one is called Buxbomidae, and fourth one is called Brydidae, and fourth one is called Brydidae, and fifth one is called Polytrichidae. These are the 5 subclass which comes under the Bryopsida. When we move into the order of each subclass, each 5 subclass, you can see in first case, Spagnidae is the subclass. Its order, it possesses single order, that is called Spagnales. Like that, Andreidae is the second subclass that also possesses one order, that is called Andreiales. And third one is called Buxbomidae. Its order is Buxbomiales. And fourth one is called Brydidae that possess Physidendales, Dichronales, Potiales, Grimiales, Schizostigale, Tetraphyllales, Ubriale, Isobriale, Hookeriales, Hypnobriales. So that Brydidae possess 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 order. And when we move into the last fifth subclass, that is called Polytrichidae, that possess 2 order. One is called Polytrichales and second one is called Dosoniales. So this is the brief notes about the Rothmeiler and Proskar system of classification. So when we move into the salient features of this Hepaticopsida, Anthocerotopsida, and mosses, you can see some similarities and some dissimilarities. Actually, this classification of bryophyte based on their similarity and difference, and it also recognized by ICBN. And later, this classification was adopted by different scientists like Taktajan, Schutzer, Parihar, and Udar. So this is about the classification of Rothmeiler and Proskar. When we move into the salient features of this classification, you can see the first class, that is hepatic opsida. That hepatic opsida include liver, that include Rixia, Markantia, Lunularia, Targionia, etc. So such class, that is hepatic opsida, when we move into the salient features of this hepatic opsida, you can see its plant body is called thallus. That plant body possesses both the dorsal and ventral surface. And some members of this hepatic opsida, the thallus of some members of this hepatic opsida are the thallus look like in the form of a leafy. So that's why we can say that in some members, the thallus is foliose in nature, that is leafy in nature. In some cases, the thallus look like the strap, that is strap of a watch, like that. And we know that that thallus shows a prominent dichotomous branching also. I think in degree classes you had studied Rixia, Markenshaw, etc. Such kind of bryophytes comes under this category, that is hepatic opsida. When we move into the root system, you can see hepatic opsida possess tuberculate rhizoid or smooth-walled rhizoid, and their ventral surface also possess multicellular ventral scale also. And when we move into the cellular organization, the upper surface, when we move into the anatomy of the thallus, the upper surface possesses filamentous photosynthetic region, and lower portion possesses storage region. So the thallus shows a distinct differentiation into upper photosynthetic and lower storage region, and that photosynthetic region possesses photosynthetic cells with chloroplast, and within that chloroplast we can see pyrenoids. Pyrenoid is a special structure found within the chloroplast, and that pyrenoid possesses starch, proteins, etc. And like that, when we move into the anatomy, its storage region also possesses oil bodies. We know that oil is a form of energy product. The oil plants store food material in the form of starch, in the form of protein, and also in the form of oil. So oil bodies can also found within the storage region. And when we move into the sporophytes are usually reside or live within the thallus or within the gametophyte. So its sporophyte is completely dependent the gametophyte for its survival, and that sporophyte possesses food, seta, and capsule. And when moving into the development of this sporophyte, the sporogenous tissue is usually formed from the endothecium of the early embryonic stage. And columella and elatus are absent in case of hepatic opsida. When we analyze the structure of sporophyte, we cannot be able to see the columella and elatus in hepatic opsida. That is why columella and elatus are absent in the sporophyte of hepatic opsida. So this is the main point related to hepatic opsida. When we move into the anthocerotopsida, this class possesses only single order, that is anthocerotiles. We already discussed in first slide. And when we move into the salient features of this anthocerotopsida, the plant body is thallus and dorsiventral, similar to that of hepatic opsida. But here, when we analyze or view the anatomy of the thallus, we cannot able to see cellular differentiation, or we cannot able to see upper photosynthetic and lower storage region. The whole part is homogeneous in nature. But in case of anthocerotopsida, ventral portion or lower portion lack the scales, multicellular scale, but it is present in case of hepatic opsida. And we can also see the chloroplast and pyrenoid in case of anthocerotopsida, like that of hepatic opsida. And sex organs are embedded, they are buried or embedded. Sex organs are usually antheridium and archegodium. They are embedded or buried within the thallus. And after fertilization, that sex organs produces sporophyte, and that sporophyte actually present within the thallus, but actually they are located on the upper surface or dorsal surface of the dorsal part of the thallus, but they from that dorsal part they emerge aerially towards upside. So we can see the presence of sporophyte at the aerial part of the plant. In simple terms, we can say that they originate from the upper surface and move aerially towards outside. And columella and pseudo-elaters are absent in case of Anthoceratopsida. Foot and capsule in case of Anthoceratopsida, foot and capsule are absent. Foot and capsule are absent. And also, foot and capsule are present. Sporogenous cells are usually arising from the amphithecium. But here, foot and capsule are present. Seta is not well developed. In case of Anthocerata, foot and capsule are present. Seta is not well developed. When we move into the Bryopsida, that Bryopsida is also called mosses. So it is somewhat different from the first two class. In Bryopsida, plant body is called gametophyte. Plant body possesses gametophyte and sporophyte like that of the previous two. But here, this gametophyte is divided into two stages. The gametophyte shows two stages. One stage is called protonema stage and second stage is called leafy stage. Actually, the protonema stage started to develop from the spore. By the germination of the spore, then start the protonema. Spore germinate and form a multicellular horizontally growing green filamentous structure which grows horizontally over the substratum. That is called protonema. It is divided into primary and secondary protonema. And from that protonema, erect stage having leaf-like structure, stem-like structure, root-like structure develops. That stage having root-like, leaf-like and stem-like and erect form is called leafy stage. And when we move into the sporophyte, sporophytes are usually developed from the leafy stage, and that sporophyte process well developed food, seta and capsule, but columella is absent. And when we move into the capsule of that Bryopsida, you can see that capsule are highly developed. The capsule wall possesses stomata, and that capsule possesses a special structure called operculum, peristome, etc. In case of this Bryopsida, also elaters are completely absent, but elaters are present in case of Anthoceratopsida. That elaters help for the seed dispersal. But in case of Hepaticopsida, when we take the case of Tardionia bryophyte, in that cases we can see elaters, but it is a rare case which comes under the category of Hepaticopsida. So this is the brief classification about the Bryophyte based on Rothmeiler and Proskauer system of classification. We'll study all the salient features of this Hepaticopsida, Bryopsida and Anthoceratopsida in detail when we move into the case study. So at that time you'll get more and more clear idea about this Hepaticopsida, Anthoceratopsida and Bryopsida. Okay, thank you. We'll discuss type study in next class.